A dinosaur bone that has been buried for 66 million years just tested positive for its own original protein. Researchers at the University of Liverpool ran a 22-kilogram hip bone from a duck-billed Edmontosaurus, dug out of South Dakota's Hell Creek Formation, through a mass spectrometer, and it came back holding fragments of collagen, the structural protein that gives bone, skin, and tendons their shape. Collagen is not supposed to survive that long. The chemistry says the bonds holding a protein together fall apart within a few hundred years once water reaches them. Not 66 million. For 3 decades, every time a paleontologist claimed to find soft, organic material inside a dinosaur fossil, other scientists accused them of contaminating the sample with modern bacteria, soil, or their own skin oil. This time the team used a more precise test, hunting for one amino acid that shows up almost nowhere except decayed collagen. And it showed up anyway. Today we look at what they actually found. How a protein could possibly outlast the rock around it. Why the contamination fight has run for 30 years. And where this evidence still falls short of settled fact. The specimen is a sacrum, the fused block of vertebrae at the base of the spine, pulled from an Edmontosaurus, a duck-billed plant eater that graced North America right up until the asteroid impact that ended the age of dinosaurs. At 22 kilograms, roughly the weight of a large dog, it is a real chunk of skeleton, not a fragment, which mattered for the test the team wanted to run. Instead of just checking for any organic residue, the Liverpool researchers used tandem mass spectrometry, a method that weighs and breaks apart molecules to identify them by exact chemical signature, to hunt specifically for hydroxyproline, an amino acid that forms almost exclusively when collagen breaks down. Finding an organic compound is one thing. Finding one that only exists as a byproduct of a single specific protein is much harder to explain. Away is contamination from soil bacteria or a lab technician's fingerprint. Scientists at UCLA independently confirmed the readings. And the Liverpool team checked the fossil under polarized light microscopy to make sure the structures actually looked like preserved tissue rather than mineral buildup that had simply formed the same shape. Every added layer of testing aimed at the same target, ruling out every explanation except the protein being real. A normal peptide bond, the chemical link that strings amino acids into a protein, has a half-life of about 500 years once it is exposed to water. Run that forward and collagen should not exist past the first few thousand years of burial, let alone 66 million. Chemists at MIT think they found the loophole. And it is not dehydration or mineral replacement. The two explanations paleontologists leaned on for years. It is something happening at the atomic level, inside the bond itself. In a collagen molecule, the electrons around one peptide bond appear to reach into the neighboring bond and block water molecules from wedging in and breaking it apart. A built-in seal running the length of the protein. Collagen is also built entirely from a triple helix. 3 strands wound around each other with no weak, unprotected section, so that seal repeats over and over with no gap for water to exploit. Put together, the mechanism does not make collagen indestructible. It makes it degrade in slow motion instead of falling apart in a few centuries. That lines up with what the fossil record is now showing. Not full, intact protein, but persistent fragments, worn down yet still chemically identifiable millions of years later. This is not the first time someone has claimed to find soft tissue inside a dinosaur bone. In 2005, paleontologist Mary Schweitzer reported flexible, stretchy tissue in possible blood vessel structures inside a Tyrannosaurus rex bone. And the finding set off a fight that has never fully ended. Critics argued the material was almost certainly modern contamination. Bacterial biofilm that had colonized the fossil after excavation. Or residue left behind by the researchers handling it. Because nothing in known chemistry explained how organic tissue could survive tens of millions of years underground. Schweitzer and her collaborators spent years defending the find, refining their methods, and publishing more evidence. And the debate became one of the more bitter ongoing disputes in paleontology. What makes the Edmontosaurus result different is not that it is the first claim. It is that it targets a chemical signature contamination struggles to fake. Bacterial and skin oil do not reliably produce hydroxyproline in the pattern collagen breakdown does. And the independent labs involved were specifically trying to rule out the explanations that sank. Earlier claims 30 years of skepticism did not go away. It forced the evidence to get better. Even if every result holds up. This is not intact protein and it is nowhere close to DNA. What the mass spectrometer found were short, degraded fragments of collagen, chemical leftovers, not a working molecule you could sequence like a genome or use to clone anything. Dinosaur DNA specifically breaks down on a scale of thousands, not millions, of years. So nothing here changes that. The sample size is also one bone from one animal, buried in unusually favorable conditions in the Hell Creek Formation. Extraordinary preservation like this appears to require a rare combination of rapid burial and mineral chemistry that most fossils never get. So this is not evidence that protein commonly survives in dinosaur bones generally. And the field's own history is a reason for patience rather than certainty. Multiple past soft tissue claims looked convincing at the time of publication and still faced years of challenges before, or instead of, being accepted. The hydroxyproline test is a genuinely stronger method than what came before it. But real confirmation will come from independent labs outside this collaboration replicating a result on separate specimens, not from one paper. However careful. Recap. A 66-million-year-old Edmontosaurus hipbone tested positive for hydroxyproline, an amino acid produced almost exclusively by decaying collagen, using a more targeted method than earlier soft tissue claims and cross-checked by labs at Liverpool and UCLA. A newly proposed chemical mechanism involving how electrons shield collagen's peptide bonds from water, offers a real explanation for how any of this is even possible. Signal or noise? Mostly signal. The testing method is more rigorous than what fuels 30 years of contamination arguments. And it targets a molecule contamination does not easily fake. Where this shades toward noise is any headline implying dinosaur DNA cloning, or a living protein just got found. What actually survived is chemical debris, not a usable blueprint. And one exceptional bone is not proof this is common. The honest version. Paleontology's most contested claim just got its best evidence yet. And the next real test is whether other labs, on other bones, find the same thing. Subscribe for the science stories that survive the fact check. Signal from the noise. Every week.